Hi,
I'm Josh, and today we're going to be talking about the phase of pyruvate that happens after glycolysis. Okay, so we know in aerobic respiration how pyruvate is made after glycolysis, like we just said, and it undergoes the TCA cycle, also known as the Krebs cycle or the citric acid cycle, and then eventually oxidative phosphorylation, and that creates 30 ATPs. So that's a really efficient process for energy, but that's only if you have oxygen. So anaerobic organisms, like if you live in the soil or other situations like that without as much oxygen, need to be able to get energy too. So they have a process called anaerobic respiration, and that only makes 2 ATPs. So it's pretty inefficient, but without it, we wouldn't have this. Alcohol!
and we even have some pumpkin ale for those whose pH is over 7.
So, we made sure to get PBR for all the Rutgers students. Anyway, so actually, that's what it is. All these things, our awesome favorite drinks, are made via this process, alcoholic fermentation.
And without this process, we wouldn't have beer and other types of alcohol. So, today we're going to be showing you how to make some hard cider. The first part of making cider is you have to gather your ingredients and equipment. So today we have yeast, and this is just a plain white wine yeast. Then we have some brown sugar, and then we have our sanitizing spray to limit bacterial growth. Then we have some apple juice, and our last piece of equipment is our primary fermenter and the airlock system. The airlock system is filled with sanitizing spray and some whiskey. First step is to sanitize all your equipment. So here's the bottle. You just want to get the spray in, get it all around, just like shake it. You want to make sure that the bottle is well sanitized. The sanitizing spray that I'm using, the yeast can digest it and convert it into sugar, so we don't have to spill it out. But if you're using like a bleach or something, you would definitely want to get rid of that before you put in your apple juice. So we also want to sanitize the airlock system and make sure nothing gets on there. And also, the table is also very important to sanitize. And
hey, I'm cutting coupons!
And so you just want to make sure everything's well sanitized. So before we start fermenting in the primary fermenter, we have to test to make sure the yeast is alive and functioning. So to test this, we put some of the yeast in a glass jar with some apple juice and some sugar. And the yeast, we can tell if it's alive by the formation of bubbles on the surface of the glass. This is due to the yeast breaking down the sugar in the apple juice and the brown sugar.
So, the yeast is actually a fungal eukaryotic microorganism called Saccharomyces cerevisiae.
Ah, there's a fungus among us!
So, what's happening is, it's metabolizing the glucose, and the process of glycolysis generates a pyruvate, 2 ATPs, and 2 molecules of NADH. And so that's going to be important for later, that NADH. So right now, the pyruvate then it reacts with an enzyme called pyruvate decarboxylase and turns into another molecule, an intermediate in this reaction, called acetaldehyde. And the pyruvate decarboxylase itself, what it does is it catalyzes the breaking off of a carbon dioxide molecule from the pyruvate, and that actually generates the CO2, which you're going to see here as the bubbles that are forming in this. So that's actually carbon dioxide gas, and that's where the carbonation comes from.
Fermentation process. So first we're going to take off the airlock. So now we're pouring the apple juice into the fermenter. I get extra points if I don't spill any of it.
Don't try this at home, kids.
This is only for skilled professionals and bored food science majors.
I don't think I could do that without spilling.
Okay, now we have some sugar. We made some sugar and apple juice solution because, as we said before, the higher the sugar content of the apple juice, the more alcohol that will be formed. So we added about a cup of sugar into about a cup of apple juice. So now we're going to pour that in here. And now we're going to pour in the secret and magic ingredient, the yeast. So now we're just about done. we have to put the airlock on. Thank you, Josh.
It's not contaminated, I promise.
It's not contaminated.
No need to spray me again.
And you want to make sure it's really in there well, because you really don't want any oxygen to get in. And the point of the airlock is to prevent oxygen from coming in. The yeast is going to start bubbling when it starts to break down the sugar, and the carbon dioxide is going to go through the airlock and escape through the holes on the top, but no new oxygen will get into the fermenter. So that's the point of it, and you want that to keep out foreign microorganisms. Mix it up, and just mix it up, make sure the yeast is well distributed. And now we just wait and watch the magic happen.
I hope this makes it go longer, quicker, shorter. Oh.
Biologically active amino acids.
Glutamate synthesis.
It's taking forever.
Just wait, it's gonna happen soon.
Okay, so while we're actually waiting for this, let's go into the rest of what's going on in alcoholic fermentation. So we left off where pyruvate decarboxylase had just turned pyruvate into acetaldehyde. So now the acetaldehyde intermediate is just chilling there. No, not really. There's another enzyme that Saccharomyces cerevisiae has, and that's called alcohol dehydrogenase. And so alcohol dehydrogenase catalyzes the conversion of acetaldehyde to ethyl alcohol, which is what the final product is going to be. So that happens when there's two NADH molecules that are formed as the products of glycolysis from what we were talking about before, and one NADH reacts with this whole, like with acetaldehyde and everything, and pretty much that gets oxidized to become NAD+, and acetaldehyde gets reduced to become ethyl alcohol. And through that whole process, the NAD pluses are actually get to be recycled and can be used in glycolysis and whenever that happens again, which is why you can add more and more sugar and it'll just keep becoming more and more alcoholic without worrying about running out of products so quickly.
But you have to worry if you make it. There is a point where if you make it too alcoholic, the yeast will die off. So it depends on the strain of yeast that you're using. So like wine yeast can they can support more alcohol. They can still grow in a higher alcoholic environment, whereas beers can't. So that's why it really depends on the strain of yeast you have. Or you could try using the force.
Yeah.
I think that works too.
Yeah, 50-50.
So after the fermentation was finished, we put it into bottles. Here's what it looks like.
It's gonna be awesome. Whoa, bubbles.
And
It's a good fermentation job right there.
here it is. Cheers.
Product placement.